Thank you for joining us for the first research exchange seminar of the fall semester. I'm Camille Crittenden. I'm Deputy Director of Citrus and also direct the Data and Democracy Initiative here. We're really delighted to have you here, and I want to welcome also our remote viewers. We are webcasting this live also to our sister campuses at Davis, Merced, and Santa Cruz. Help yourself to lunch if you have signed up for it. We're going to continue the practice of registering for these events this semester and throughout the year so that we can get a more accurate count for sandwiches and other refreshments. But if you have registered, please feel free to help yourself. There's water also in the back and the usual compost, recycle bins, etc. Before I introduce our distinguished speaker this afternoon, I just want to call your attention to a couple of other upcoming events that I'm very excited about. One is being co-sponsored by the Data and Democracy Initiative and the UC Berkeley Institute of Governmental Studies. The question is, can open data improve democratic governance? This will be held next Thursday and will include a keynote address by California Lieutenant Governor Gavin Newsom. We'll have another keynote address by an IBM executive and then several panels that will address the topic of public open data. What are the opportunities? What are the challenges? What are the ethical considerations that we should be thinking about by having all of this personal data now made public? Another event that's coming up shortly after that is a special distinguished lecture by Jaron Lanier. I don't know if you know his name. He's the author of You Are Not a Gadget, and more recently, Who Owns the Future? He has wide and varied interests in music and virtual reality and other instances of computer technology innovation, and he has really interesting things to say. The provocative title of his talk is How Can We Prevent Information Technology from Destroying the Middle Class? So please do sign up for that quickly because it's actually on its way to selling out. It's free, but we do ask for your registration. Lastly, I want to announce the Big Ideas competition. You may know about this competition that is run through the Blum Center, focused on student teams that have big ideas for improving humanity in all kinds of ways. There are different categories. Citrus sponsors a category on information technology for the betterment of society, so that's one. The Data and Democracy Initiative this year is sponsoring two categories, one in human rights and technology, which we did last year, and another that's a new category related to the conference we're holding next week, actually, on open data. So if you have students or if you are a student, you know students who are interested in pitching ideas to this Big Ideas competition, there's an informational talk tonight, actually, at 6 o'clock in Blum Hall, which is just across the way there. And the pre-proposals are due November 4th, so be sure to put that on your calendar and take a look at the information that's available about that. So I'm pleased to introduce our speaker this afternoon, Patrice Koehl. Patrice is currently a professor in the Department of Computer Science as well as the Associate Director in Bioinformatics at the Genome Center at University of California at Davis. He received his PhD in molecular biology and biophysics at the Université Louis Pasteur of Strasbourg, France in 1989. The same year he was appointed staff scientist of CNRS and joined the biological NMR laboratory at the same university. In 1997, he came for a sabbatical year to Stanford and liked California so much that he decided to stay. In 2004, he joined UC Davis with a joint appointment as professor in computer science and at the Genome Center. So please join me in welcoming Dr. Patrice Koehl.
Thank you. Thank you for the really kind introduction. It's a great pleasure for me to be in Berkeley. Something she did not mention, the first time I came to Berkeley was 22 years ago. I'm ashamed to admit so many years. And I spent 2 years here, and this was something that hooked me to California. And it's going to be very difficult for me to even think of leaving California. What I'm going to try to do today is to give you a lot of data. My talk is about big data, so why not give you data? But the purpose of my talk is to go through this data and hopefully show you that in data there is information, and that from that information we can go into knowledge, we can go into understanding. And I'm going to follow a scheme where I'm first going to educate you. Seems strange because I have sure many people in the audience are better informed about big data than I am. But I'm going to try to inform you in a slightly different way about what big data are. And I'm going to start with something that I find, at least I found when first time I came to Berkeley, is a characteristics of the English language in the US, which is the use of acronyms. Remember arriving in Berkeley and saying, "Where is UCLA?" And people looked at me not understanding that I was speaking about UCLA. So let's use acronyms. For me, the best way to represent big data is to use the acronym III. Why III? Because what big data is? Big data is first ill-defined. There is no unique definition of what big data means. So we'll go through that and we'll try to understand what really it means. Big data is immediate. We need to do something yesterday, not in 2 or 3 years from now. Big data is now. But big data is also intimidating. That's the third I. What if we don't do anything? What if we ignore the wave and don't cope with it? Are we going to be in the same position as if we had tried to do something? So let me start with this concept of ill-defined. First, a little bit of vocabulary. A data is usually assimilated to a storage on a computer. There is a unit of measure for data. The basic unit of measure for data is the byte. So the byte is a row of 8 containers that can have the value 0 or 1. A byte doesn't store much. So when we speak about data, we speak about powers of bytes. It's very common to speak about 1,000 bytes, kilobytes, megabytes, gigabytes, terabytes, petabytes. This is where we have to learn our vocabulary, or go back to school and learn our vocabulary again. Exabyte, zettabyte, yottabyte. But if I just give words and don't try to relate to something we know, we are a little bit lost. So what are these words related to? As I said, the byte is the unit for measuring the size of data. Kilobyte is approximately the space you need to store a page of text with nothing fancy in it. Megabyte, that's the space you need to store a song. Gigabyte, we move forward, we go to the movies. Terabyte, it's a little bit harder to represent what a terabyte is, but it's equivalent to 6 million books, a very large library. Fits in a terabyte. Terabyte is the current size of the common hard drive that you can find on computers. What's a petabyte? Well, if a terabyte is already 6 million books, a petabyte would be 6 billion books. Or a better way to represent it is take a building of 55 stories and just accumulate DVDs there. That's the space that a petabyte represents. Exabyte is the estimated amount of information that was generated up to 2003 from the beginning of history. Zettabyte which is 1,000 exabytes, is the amount of data that was generated in 2011 alone. So notice the difference. The exabyte was up to 2003. The zettabyte is 2011 alone. Yottabyte, 1,000 times the zettabyte. That's the size of the big data center that the NSA is building next to Salt Lake City that will be open and running by the end of the year, getting us in even more trouble with foreign nations. Now, even though I'm a computer scientist, it's hard for me to see those scales. So what I usually like to do is use metaphors to represent them. If you take the byte as 1 second, the kilobyte is already 20 minutes. The megabyte is 11 days. The gigabyte is 30 years. The terabyte is 300 centuries. The petabyte is 30 million years, longer than humanity's. The exabyte is 30 billion years, older than our universe. And I'm not reaching the yottabyte yet, the level of the NSA. So this gives you a scale of what data can be. And this gives us a scale of what data are today. NSA, this is not in the future. This is 2 months from now. But big data is not simply volume. It's not simply size. Big data is also velocity. We are now reaching a stage where the information transfer, now I'm not speaking about the amount of information, but I'm speaking about the transfer of the information, reaches speeds that were unknown even a few years ago. In 1 minute, 204 million emails approximately are sent. In 1 minute. Over the time of my talk, more than 640 terabytes of data will be transferred on the internet. 6 million users will be connected on Facebook. Just 1 minute of time. 1.3 million videos will be watched during that 1 minute. More than 3 million Google search happen. So big data here relates not just to the speed, to the size. Each email is usually very small. But big data is now related to the velocity. You have 204 million of them going around on the internet during that time. But big data is also diversity. Big data is not just text. It can be numbers, it can be images, it can be sounds. All of these are stored in the format of these bytes, series of small containers with zeros and ones. And that leads to my second acronym for today. So you'll get out with a few acronyms. After the III, I'm here at the VVV, volume, velocity, variety for big data. But that's not the only facet that we need to think about when we think about big data. This volume velocity is one thing. Variety relates to, as I said, images, sound, numbers, tables. But it's also some data are structured. That's what scientists deal with all the time. Some data are unstructured. The emails that I send around are unstructured. There's no, per se, rationale behind them. It's just that 200 million people have sent an email. We don't know what the content is. We don't know how this is organized. But really, I prefer to forget about size. I prefer to forget about this velocity. I prefer to take all that under one umbrella term, which is complexity. Complexity means that we are dealing with a system that go beyond our capabilities of getting the information right away out of it. When I started working with computers, big data was in the million bytes, in the megabytes. Programming on a computer where you had only kilobytes of memory was something common. And when someone would come to you and says, I have this matrix of size 1,000 by 1,000, you get worried. Today, people don't even think about it. But the complexity of the problems that we're dealing with did not shrink. In fact, increased. For statisticians, if there are some statisticians here, we are speaking of large N, large P problem. What does that mean if I translate that in sort of common languages? Large N means large number of data points. We have them. But large P means a large number of variables that are responsible for the signal we observe in this data. And the more we have data, the more we hope that we are able to find the meaning of this variable and find their influence on the data. And what used to be hidden variables that we are not able to deal with becomes variables that we are able to take as integral part of the model that we define for the system. Big data defines a problem of security. We all already confronted with that. It's nothing to do with big data. I don't know how you feel, but I still feel insecure when I connect to my bank account through the internet. There's a question of security here. Now, what about things that are this, as much as this is very pertinent to me, what about other information that translates into numbers that go through the internet? How do we find security in sending them? Big data, now it relates to the problem of volume and velocity, raise questions and challenges for integrity. We are transferring data at a rate where there's a risk of failure. How do we deal with these risks of failure? What about the integrity? What about the validity? The more you have data, the more you need to ask yourself the question of, are these data valid? Are they really represented the system that I'm interested in? How much trust can I put in the source of these data? But big data is not just challenges. Big data creates opportunity. Probably the easiest opportunity to mention is something that has become very popular in the literature in general. It's this concept of fourth paradigm for scientific research. The fact that we are no more, or in parallel to an hypothesis-driven type of science, we move towards a data-driven science. We do not anymore only rely on questions that we ask and then check with the data if the data validate or invalidate any hypothesis we make on a system. We let the data tell us something about the system. We move from the data and we hopefully extract from this data information that can then be translated into knowledge, which itself hopefully leads to societal benefits. So this is the fourth paradigm. No more hypothesis-driven science, but a science where the data are telling us what to look for. We are, in some sense, the more open-minded you are, the less you limit yourself to a few hypotheses you have, the better you'll dwell when you deal with large amount of data. Big data provides another opportunity, something I call a holistic approach to major research effort. We are no more limiting ourselves to small segments of a research project. We are now, you can see now in terms of the efforts that are put forward for funding, you can see projects that go from the minuscule to the large, very large scale. The Human Brain Project that was funded last year by the European Union at the level of $1.3 billion is to go from the single neuron to cognition. We are having a holistic view of what the brain is, and that's entitled by big data. Big data offers new paradigm in computing, a different way of thinking, how we put together instruction for solving information, because the data are there. Big data offers huge opportunities in digital humanities. There are wonderful projects going on in many universities in the US about digitalizing all fiction books that have been written since they have been in press. How much information are we going to get from that? How much plagiarism are we going to find between writers? We all ask our students to use turnitin.com to see if they plagiarize. Did the authors in the 1900s plagiarize also? So, I mean, this is information that we're going to learn a lot about the system that we have. I don't like the word opportunity. When I speak about big data, I want, in some sense, you to feel that big data is something that will enable dreams, something that will allow us to move forward to dimensions that goes beyond just the research project we finished by the end of the month, something that goes beyond what a single person can do. What type of dreams people are looking at when they look at big data? In physics, the physics of dark energy is probably the biggest effort that people are speaking about. How the brain works, I was just talking about it, from the neurons to cognition. How do we go from the isolated billions of neurons that we have into a system that provide cognition? How do we connect the infinitesimally small to the large scale and to the function? A holistic view of natural ecosystems with all the sensors we have everywhere. We do not just have a view based on a single camera or a single system recording what's happening at one point. We have an overview of the ecosystem at large. So we have a way to dream about understanding how ecosystems work. Understanding climate changes. We all know climate change. Do we know how they change? How do we get the data to learn how to change? Well, the sensors are there. We just have to come up with a picture and put it together. From genotype to phenotype. What makes us different from one to another? I'm going to speak a lot more about that in 10 minutes, but you'll see that we have the tools to go from the blueprint that represents us to an understanding of the effect of that blueprint. Precision medicine, this is another big thing. Precision here includes individualized medicine, personalized medicine. the ability to treat a person according to not the statistics on the population, but on the effective illness and the effective system that the person represents. So here we have a way to go beyond what current medicine is doing. Big humanities, digital history, digital literature, digital world, where you start learning about how the psychology of digital world could represent and what it tells us about who we are. So that's what big data is. When I said ill-defined, I meant basically the message I just want you to get is big data is not just volume. Big data is complexity with a general meaning of the word complexity. Big data is a source of challenges, but big data is a source of dreams that allow us to think beyond the everyday problems that we usually deal with. Now let me come to the second part of my talk. Now that I have defined big data, let's see how immediate it is. Let's see how it actually affects us in research. And I'm going to use one example for that, which is close to my own interests, which is genomics. And I'm going to lead you through some of the data that are currently available in genomics and what they mean and what they could represent in terms of future. So genomics has the word genome in it. So let me just briefly refresh you on what's this genome. You all know that in a cell, you have a nucleus that contains the blueprint of who we are. This blueprint is, in fact, a very long molecule. If I was to untangle that molecule that we have in our nucleus, it reaches 1 meter long, approximately. And that molecule is called DNA. And what is that molecule? That molecule is a signal. It's a set of letters, A, T, G, or C. How many letters do we have? Since we have 1 meter long of DNA, we have space to put a lot of letters. And how many we have? We have approximately 3 billion letters sitting there. And these letters, just like letters in the English alphabet, have meanings. They don't have a meaning by themselves. They have a meaning when you put them together in words. It's just like English letters. When you put 3 letters together, depending on which letter you pick, it may mean something, but if you just pick random letters, you may not get a word that means something. Well, it's the same thing here. Among these 3 billion letters, there are regions that really have a meaning. These regions that have a meaning are usually referred to as genes. And a gene is then used as a support of information. And then what the cell does is it just, when it needs the product of that information, it just goes there, reads the information, and translates that information into and generate a molecule, and that molecule is a protein. And these proteins that we have sitting around in our cells, they are the blue-collar workers of the cell. They do everything. They clean up, they make sure that the cell breathes, they make sure that the cell eats, they make sure that the cell eliminates. It performs all the heavy-duty tasks that the cell needs in order to survive. But sometimes this coding information may get distorted. It may not work anymore. It may not perform the task it was designed for. Genetic disease are just that, the result of a message contained in a gene that does not perform its task as it's supposed to, because the message was changed. How do we understand how we get from the message to the product, and how that modification of the message can lead to modification of the function, is a task that people have been trying to solve for a long time. There's many studies that have started more than 100 years ago on trying to decipher that. Nearly 100 years ago, it was shown that the genetic code was carried by the DNA. Then it was shown that the DNA is transcribed into RNA, translated into proteins, and these proteins are the responsible tools for the machinery of life. So people said, if we want to understand what's happening here, let's get the message. Let's get the DNA. That's all the genomic studies, that's what they do. We can look at the transcribed region and forget about the region that do not code. That's looking at the transcriptomics, all this omics project that you may have heard of in the literature. What about the protein themselves? That's the proteomics project. What about regulation? Regulation comes from the fact that proteins interact with each other. And when proteins interact with each other, that's what generates their function. So if we want to understand how they work, we need to understand how they interact. That's with the omics. If we add it at the end, it's interactomics. Metabolism, how we go about the different substrate that enter and leave the cell. That's metabolomics. 3 billion letters. As I was introduced, I did a PhD in molecular biology, and then I'm not ashamed to say it was around in 1984. And at that time, I had to do a lot of sequencing of genes. It took me a day to sequence 300 letters. So if I wanted to generate 3 billion letters, I would not be done yet. So they had two options. One is to get 300 million graduate students and ask them to do it, or find a new technology. Make it a challenge to deal with sequencing the information that we have. And in the early 1990s, that's what the US government put forward. The challenge of sequencing the whole human genome, the 3 billion letters that comes in our DNA. And it's very interesting to go back to early 1990s and to see how that human genome project scaled compared to other projects of the time. The big project of the early 90s was the Space Station Freedom Project. Estimated cost $30 billion. Well, it never got realized. It was replaced by the International Space Station because the cost would have been much higher than the $30 billion that was put forward. What about, let's speak about the SSC. $11 billion was abandoned also because of costs. Hubble Space Telescope, the $1.5 billion turned out to be at least a factor of 10 underestimate of the real cost of putting it together. Interestingly, the only project in this list that was completed under budget and before it was estimated that it'd be finished is the Human Genome Project. It wasn't finished in 2005 as it was estimated in the early 1990s. It was finished in, it depends on which date you put. But I would say 2001 is a reasonable date. And the total cost for that project was under $3 billion. So here we are, we put a big project together, and here we finally have a project that works. How did they do it? Sequencing 3 billion letters through a single machine and expecting that you get the right answer out of it is out of the question. So what you do is you take multiple copies, you break it in small pieces, and in some sense you run it in parallel. You sequence all the small pieces, not with 300 million graduate students, but with multiple tracks on the sequencer. And then you put all the information back together because you have overlap between the pieces that you have that allows you to reconstruct the signal. And this is still the technology that is used today. But really, where you see the impact when you put the pressure on solving a problem, you see that the technology follows. The technology we're using in the 80s was designed for the problem we were dealing with. We were happy with sequencing 300 letters. That's the only thing we needed. So we never thought of looking bigger than that. So we never thought of improving the technology. When it was a question of sequencing 3 billion letters, then the technology was improved. And if you look at the cost per 1 million letters, well, in around 2005, it was still $10,000 to get 10 million letters. So to get to the 3 billion letters, that was around the $3 billion I was mentioning. Today, it's 10 cents. Today is 10 cents. The improvement that we see here went faster than what would have been expected based on a famous law in computer science called Moore's law. Moore's law just says that every year the power of computers increased by a factor of 2. So if you look at how much computers have improved since the early 1960s, when they started having more computers, you have that exponential increase in power. But that exponential increase in power is orders of magnitude smaller than the decrease in price that we observed for sequencing a human genome. Sequencing a human genome was around $100 million in 2005. Today, the numbers here are wrong because it's less than $10,000 to sequence a human genome. We're close to $1,000 for sequencing a human genome. And if we keep on going like that, in 3 to 4 years from now, it'll be around $10 to sequence a human genome. That means it's going to be a cost of $90 billion to sequence the genome of all humans. Think of the number of data that will generate. Think of the size here. I like to represent this slide a slightly different way. So this is just an increase in the number of sequences that we have, and the red part is just coming from this whole genome sequencing. But I like to show you that slide. It looks complicated, but it's very simple. Let's look at growth as a function of time. And let's look at the reference, which is Moore's law. Here, that's the red increase that comes from this every year the power of computers increased by a factor of 2. Let's compare that with other growth. World population, hopefully, it's a good thing, doesn't grow as Moore's law. We see a much more linear growth in the human population. Number of publications published, it increased. We have many more scientists and many more scientists that rely on publication to get their grant renewed. So you see an increase in the number of publications. The more it's going to be, the harder it's going to be to get grants, the more people are going to publish. Remember, though, one message I said before: information is not knowledge. Because that's data here. But now we look at the number of patients whose genome has been sequenced completely. That number increased at a rate that's much larger than the rate we observe for Moore's law. There are projects going on in many hospitals today where every new patient, and fortunately cancer patient, has his genome fully sequenced so that we can get a full spectrum of information about what are the causes of the type of cancers that they have and the diversities that are observed in population. What made all that possible? Well, technology. An Illumina sequencer is able to sequence a whole human genome today for less than $4,000 in 2 weeks, approximately. That's not being in a rush to do it. I like the quote by the CEO of the company that put this technology together. A single lab with one HiSeq, one of these instruments today, is able to generate more sequences, more string of letters, than was in GeneBank in 2009, every 4 days. So a single lab in 4 days produced more knowledge than was available in 2009. Just need to put some advertisement for my own campus. At UC Davis, we have a genome center where we have 3 of these machines that are sitting there, some are using other technology. We collaborate with BGI, the Beijing Genome Institute. And the Beijing Genome Institute, the outpost they have in Davis, generates 5 terabytes of data a day. That represents 1,600 human genome if we only look at one read of that human genome. And it generated 500 terabytes of data a day. This is where the BGI and the human genome connect together: data, knowledge, societal benefit. Let me skip that slide and go into something that is more relevant directly to that talk. What do we expect from this data? Well, one of the big things we expect is molecular diagnostics, looking at different ways to understand what's the pathology that the patient may have. Traditional way, especially in cancer, for cancer patients. You take a tissue sample and you look at it through the microscope. And a pathologist, by looking at the type of cells that you have and the fact that some of the cells may not look like they should, based on a library of shapes for cells, was able to provide a diagnostic for the type of pathology that was affecting the patient. The new way would be you take the blood from the patient and you analyze its DNA. And now you look at which proteins are expressed from this DNA. And are they the proteins that we expect, or do we see a difference? If we see a difference, then we know what was the functionality that has been modified in that pathology. So instead of looking at directly the end product, you can look also at the DNA level and understand how that DNA level was modified. And that provides a diagnosis that relates directly to the changes that occur in that patient. What is interesting is this slide is not the product of my imagination for what will happen in 5 years from now, what I hope to happen in 5 years from now. This slide is on the front page of the web server of the National Cancer Institute. That's their way of thinking. That's not my dream for the future. That's the current way of thinking of the National Cancer Institute. So that's the whole idea. You take some cancer cells, you isolate either the DNA or the RNA, and you analyze it. You sequence and you compare it with the sequence of regular protein. You look at the expression of this protein. You look at mutations that can have occurred. You look at the geometry of the chromosomes, if there are structural changes. You look at any changes that occurred to the signal itself without mutation. You could have some product that were added to the DNA. That's where we are going. What is fascinating, though, is $3 billion 13 years ago to sequence a human, an individual. Today, let's say $3,000. Are we saving so much money that we should ask much less money to the NIH to do our studies? No. The money is just shifting from one position to another. What used to be that we were spending more than 80% of our money into sequencing, today we're only spending a small fraction of our money into sequencing, but we're spending a huge amount of money in the analysis of this data. And this is where the big data challenge really shows up. And this is where the big data challenge shows as something immediate. Let me go back to a quote I gave you 3 slides ago. CEO of Illumina says, look, the technology we have today. With the one of our instruments, you generate more sequences that were in GenBank in 2009 every 4 days. But let's look at the reality now. I have an Illumina HiSeq. I'm not interested in a human patient. I'm interested in sequencing the whole genome of a pine tree. And I'd like to find what is the sequence of that pine tree, so that maybe I can understand why there's this population of pine trees in Northern California that are really suffering right now. And in a week, I get all the raw data coming out of the sequencer. But when do I get my actual sequence of my pine trees? Well, I have 60 billion fragments that I need to analyze. It takes me 10 days just to do error correction on those, because there is an error rate in producing that large amount of data in such a short time. It takes me 11 days for assembling big reads, because I don't have a computer that is large enough to deal with these 60 billion fragments at once. So I regroup them in what I call super reads, and I assemble these super reads first. And then it takes me another 60 days to build that full genome that I have, and another 8 days to polish up at the end and fill in the gaps. So 4 days to get the data, 3 months to get the knowledge. Get the information from this data. This is not yet knowledge. So that's where the big data challenge here is. That's where the big data challenge is immediate. That's where we need to deal with it today. Because we can't let scientists wait for 3 months to get the information out of data they generated in 4 days. This is where we need to do something today, not tomorrow. We should have done it yesterday, in fact. Let me do a side note on the social consequences of all this big data. And let me do that with respect to genomics. I always like to do some epistemology to provide us a feedback of what we learn from what we do. There's a big danger of misuse of the data that will come from the genomics project. What type of misuse? For example, predict sensitivities to various industrial or environmental agents. Could an employer use that as a criterion for hiring or not hiring someone, if that information were to become available? The impact of information that is likely to be incomplete. What does it mean if your genome tells you that you have 25% higher risk of developing cancer? I'm sure you read in the People's Press the whole story about Angelina Jolie getting mastectomy because she has an increased risk of developing cancer. How do we treat that information? There's a reversal of knowledge paradigm. I'll come back to that in just a second. Are the products of the Human Genome Project to be patented and commercialized? It's not an old debate. It was in the Supreme Court in May. It was this whole affair with Myriad Genetics and the BRCA1 and BRCA2 genes. These are two genes that are susceptible to detect a higher risk to develop breast cancer in women. And changes in these genes are related to this risk. But the technology to do that sequencing and detecting it was patented. So there are questions here. How do we educate about genetic research and its implications? How much of a epistemology lectures do we include to the education of the biology students today? How much do we educate them about what's happening in the consequence of what they are studying? So let me just show you some example is about this paradigm shift that we observe related to medicine. There's a company, 23andMe, that if you go online, you can order a swab. They'll send it to you at home. You swab, get some sample. You send it back to them. They are going to study your DNA based on that, and they're going to give you a full report about what they get. They don't sequence the whole genome, but they sequence fragments, and based on these fragments, they're going to give you some information. For example, for type 2 diabetes, they're going to tell you if you are at low risk of developing type 2 diabetes or if you're at high risk of developing type 2 diabetes. May go as high as 52%. What do you do with that information? What do you do with that information? I personally feel that the risk is even higher. This is a page that I took directly from the company, and that's the page. As much as I have nothing against 23andMe, I have something about that page. I really have something about that page. Sensitivity to warfarin. So warfarin is a common blood thinning drug that's given to people, to some patients that went through a stroke to avoid a repeat. What 23andMe will tell you is how susceptible you are to that drug and how much effect it can have on you. And they conclude with what to tell your doctor. So that means a patient, a person on the street that has not been educated in medicine is going to be more informed than the doctor, is going to provide feedback to the daughter about what to do. That's the reverse paradigm. And I'm concerned about that. Something else that's a little bit just funnier, people may start putting their DNA information on their Facebook page. And then you'll have, let's just be friends. It's my DNA, isn't it? So consequences of that information. What if we do nothing? Let me finish with that. What if we do nothing? What should we do? We don't want to penalize current research. Remember what I said about the sequencing? 90 days to get the information from just 4 days of collecting data. We need to do something about that. We're going to block new initiatives if we do that. What about the new generation of students? So how do we approach big data? I love the web page for the Human Genome Project. That was at the beginning of when they started this Human Genome Project. If you see it, it's not just molecular biology. The Human Genome Project is a project that involves people from informatics, engineering, chemistry, biology, physics, ethics. This is a global problem that needs to be dealt with an interdisciplinary approach. And my own point of view is, how do we deal with big data? Technology, and I've showed you, technology moves fast. We have Moore's law to help us with computers. You've seen how fast technology was able to sequence the genome. What is it that we are missing? We're missing the people. We're missing the people with the knowledge of how to deal with this big data. So the real solution for big data is really going to come through training. Training will allow for new discovery, will allow for new developments, and training will allow for distribution of the information that we have there. And I'm going to stop here, and just I finish by the closing sentence I like to put when I speak about big data. Big data is big dreams. They are complex. They lead to information. But we have to be aware that to get to the knowledge, we need to put the effort in understanding what information means. And I'm thanking you for your attention.
Thank you. Do you have any questions from the audience? Let me use the mic. Oh, here we go.
Very interesting discussion. The part that was a little bit surprised that you were actually concerned about the reverse effect, that a patient would let a so-called expert to tell him about the analytics or the information that he gathered more than that. So I think actually that opens up the whole new approach of training, as you were emphasizing, that now the new generation of the physicians maybe should be trained as an extra population starts catching up. So I think there's a need for the new generations of so-called experts to go back and learn new things so they could be ahead of the curve, so the general public shouldn't really teach them, so they could be a leader on teaching the regular public. And
at an even more basic level, we should train physicians to deal with patients that comes with a knowledge supposedly bigger than they have. So we really need to do a lot in training. That's, I think, the stumble block that we're going to encounter in the coming years. Is big data is not technology. That's not where the problems are. It's in training.
I had a question about the training, specifically in the area of, let's say, genomics. I was hoping to ask this question while there were still more students in the room, but I think a lot of the students have gone off probably in their next class. But just briefly, when we think about big data challenges, let's say in the life sciences, are there training approaches that you would recommend for students who are coming from computer science, how to learn about the life sciences, or vice versa if they're coming from life sciences, how to get the kind of training they need in computation so that they can actually get involved in this work in big data?
So I'll answer your question by starting with the second point. If I wanted to train a life science student to become more quantitative, the first thing I'll make sure of is that I provide a training that integrates the three aspects that is fundamental here. I do not want to maintain a curriculum where the quantitative part is taught outside of the biology. I'm too concerned by seeing so many students entering college and taking a life science major. First, because they run away from math. They have to take a calculus class, but they don't see the connection between the calculus class and what they actually need. If you integrate that into a curriculum that would, say, cover a whole year with 3 quarters, where it will be a follow-up of what you've seen, where you start with the life science, the tools that you need to analyze that, and then the application of this tool on a computer, you'll get a better understanding of why you need math, and students that are more ready to enroll in such classes. For the computer scientists, it's the same thing. The computer scientists often see life science problems as applications of what they do, and do not see the bigger picture of the life science problem being a problem by itself. It raises new challenges for computer science, but there need to be an understanding there. My call on that is I go against the flow. I'm against all online classes because the way we can solve this is by putting up teams of people working together from different fields as early as possible as freshman years in college, so that people can work together on a problem, each with their own speciality, each with their own nuance, each with their own sensitivity to the problem, so that it leads to a common solution. These common projects are already developed within departments. So you see challenges like iGEM in biology that are becoming extremely popular. We need to create these challenges at a broader scale that involves people from different fields. I think that's the solution that we need to push forward here.
I'm going to make some skeptical comments about big data, and big data may be big confusion. So last week I was hiking up in the Sierras. I had no cell phone, no computer, no TV, and my life was so much better than this week when I read the newspapers and see all the kind of stuff we get from the data. So you say that big data can lead to knowledge, but I think as we have the capability of having so much data, it becomes even more important to figure out what questions should we ask. And that's maybe an issue of wisdom. And an example, if our politicians use all the data they have on all the computers to figure out how to do as much damage as possible to the other party, rather than maybe using all the data and asking what is the best thing we can do for this country, right? Then we're having all this big data, but asking the wrong questions, we may actually be worse off than before we were suffocated in so much big data. So how do we educate our students to have this level of wisdom or common sense to figure out what are really the pertinent questions to ask, and not just grab a piece of data, looking for the answer, but answering the wrong questions?
So the answer to that question was sort of partly in my talk. I tried to cover genomics and showed how genomics is affected by big data. But remember, my last 3 slides were on the consequences of the big data we have in genomics. So questions we need to ask ourselves about what it means to have this big data. Big data, as it's represented right now, is often limited to the concept of information and doesn't go into knowledge. The real question that you ask is, how do we really put that information into good use so that it provides knowledge that itself will lead to societal benefits. The way we educate the students in doing that is we have to have epistemology, starting from freshmen. We need to have them ask themselves questions about what they do.
Yeah,
we do that all the time as scientists. We need to train them as early as possible to do that. In some sense, we educate our students, hopefully, today, to be literate when writing in English. We don't educate them too much about what they write about. We need to change that. We need to change that. And when we get there, we'll build a new generation of students that is thinking. In some sense, big data is just like any new technology. There's always at the beginning an education process that goes through that we need to go through the pain of that education process. There's an urgency here that probably did not exist with previous technology, because the speed at which we go is unseen in the last 100 years, or even before.
I'm reminded somewhat of what Heisenberg said about 50 years ago, when we were looking, what it means, quantum mechanics and the physics and all of that. He says, in the end, you're always encountering your own questions. If you ask the electron, are you a wave? The electron says, yeah, I can be a wave. If you ask the electron, are you a particle? He says, yeah, I can be a particle. So I think asking the right questions becomes ever more important now that we have in principle the tools to give us answers to any questions we ask. And so, again, I think I'm agreeing with you. There's a shift maybe in our education for the students. How to be skeptical, how to find the right data, how to make sense of it, and what questions to ask of this immense wealth of data that they can get in milliseconds now.
And interestingly, for the newest generation of students, it's not just that they can get, it's that they will get. Whoever is entering college today, in 4 years, 5 years from now, when they'll apply for jobs, will be in a situation where the employer will ask them, how quantitative are you? What's your ability to deal with data? That question will be universal, because data are universal. So we need, as educators on our side, we need to deal with that now.
I just want to thank you again for a great talk.